Good evening, everybody. Welcome to the second webinar on deep tech. Okay, it's 6:05. I suggest to wait another couple of minutes and then we can start, okay? Just as a general rule, I would say if you have any questions during the speeches, you can use the chat. The chat is, you can find the chat in the bottom part of the screen, and you write there your questions and the speakers will answer afterwards. Roberto Della Marina is with us. He will be moderating the whole seminar. And we try to keep it short and focused. So our objective is to stay around 40 minutes.
Mm-hmm. Right.
But of course, if there are many questions and so on, we can go on.
Okay, so Francesco, what do you say? Can we start or we wait?
1 minute and then we start. I'll give you the floor.
Great.
People are still coming. Now we are 18, so it's a good group, focused on the topic. Very interesting. Okay, it's 6:05, so we can start. So again, thank you everybody for coming. My name is Francesco Vanier. I'm the Associate Dean for Executive Education of MIB, and I'm in charge also of the Executive MBA in Business Innovation. That is the first MBA in Italy focusing on deep tech, entrepreneurship, and transformation, digital transformation for large companies. And actually, Lydia is one of the participants who will start the next autumn. So I'm glad to see you here. And today, Roberto, that is one of the investors of Venture Factory, the first Italian private equity and actually a venture capital fund focusing on deep tech that invested also in these startups. He will interview the two entrepreneurs, Lidia Pieri and Emiliano. They are two scientists. Emiliano is a biotechnologist and biotech, is it? Biochemistry, I think, professor in Trento.
Yeah.
And Lidia is a physicist. And I will try to stay in the background, but if something happens, I can help you in fixing anything. For the moment, I think it's all. And Roberto, do you want to introduce our guests better than I can? Than I have?
Thank you, Francesco. Thank you very much. So, hello everybody. Good evening to everybody. So, welcome to the second occasion to this deep tech stories by MIB. As Francesco has introduced the series, we are mostly focusing on deep tech cases, and looking into somehow what is deep tech, how it's declined in the different ways, in different through different companies, and example how a scientific idea where deep tech resides can propagate towards becoming a company and towards a market. So clearly deep tech is kind of new, and we can somehow understand deep tech in the business context with almost three key attributes. Potential for impact, clearly coming from scientific discoveries or significant engineering developments, need to have a potential for impact, long time to reach market-ready maturity, and substantial requirement for capital. So it somehow differentiate very much from what normally say shallow tech business can do. And shallow tech we mean web-based services or social or mobile apps and kind of this. So This is particularly suitable, I mean, this kind of a deep tech approach these days in which everybody is being affected by pandemic. And possibly I would say we see a kind of a different attitude towards science and scientific approach. Somehow has started to emerge. We definitely hope that this trend is here to stay, but we will see. So this afternoon, I have the pleasure to discuss with two distinguished guests, Lidia Pieri and Emiliano Biasini. They're coming from an extremely innovative and promising company, Sybilla Biotech, about drug discovery and especially the disruptive methods they develop. So let's get started and introducing the founder and CEO, Lidia Pieri. Hello, Lidia. Good afternoon.
Afternoon.
And Emiliano Biasini, founder and scientific advisor. Hello, Emiliano. Good afternoon.
Hi to everybody.
So, Lidia, Emiliano, two impressive CVs and two impressive somehow backgrounds. So, Lidia, you just look into your CV. You're a physicist by education. We are colleagues because you are a high energy particle physicist. Then you got your PhD in astroparticle physics. Yes. You work at CERN, I mean, international level, and in Stockholm and in Paris. So, and all of a sudden you jump out of the physics and you need to become CEO of a biotech company. This is funny. I mean, you will need to explain us a few bits in a couple of minutes. Emiliano, you, as Francesco was already introducing, you're a biologist. You earned a PhD in applied genetics, and you as well. You work at abroad in Washington and in Boston University. And a few years back, you came back to Trento as a professor at the Teleton Institute. So more or less, more in line with what the drug discovery is all about, isn't it? So Lidia, Emiliano, Lidia, starting with Lidia, can you somehow describe us a bit your trajectory? How do you move from physics, high energy particle physics, towards biology and biotech, and becoming a CEO of biotech, by the way?
Well, I must say that it's not surprising at all to me, this jump. I've always been driven by curiosity in what I was doing. When I was working at CERN, I was bridging between theory and experiment. And then when I was quitting my diploma thesis, the lab was shutting down. So I didn't want to do hardware for LHC. And I switched to another field where data were coming. And it was a very exciting moment for astroparticle physics with all the data from Fermi and AMS that were coming about dark matter. I was studying the nature of dark matter. So it was really a really flourishing period to work in. And I've always liked to bring together people with different expertises. I've joined groups which came from different areas of astroparticle, from astrophysics and theoretical physics still. So I've always been both at the borderline between different areas and inside the same area at the borderline between theory and experiment. Actually, I was called a phenomenologist because I was analyzing data, finding models, and finding a way to understand the data through the models and then to adapt to new data coming and so on. And it's pretty much what I'm doing here as a CEO. It's still a very feeding environment. So I must say, I can't live with no challenges, and this is a real challenge. So things always move on, always change, and I have to stay always on the edge. And this is exactly what I want to do, what I've done in physics and what I'm doing here. So no surprise, not so much surprise to me.
nice to hear. And Emiliano, can you discuss about what about leaving the comfort zone of being somehow fundamental, staying with fundamental research, doing class teaching, a prestigious lab, and somehow jumping in a fast-pacing startups and in this fuzzy world of business?
Well, first of all, Roberto, let me thank you all for the invitation. This is actually a great occasion to, tell you about my history and my involvement in Sybilla. I guess is the perfect venue. So first, to answer your question, everything has to be got back to my experience in the US. I spent almost 10 years in the US, and I must admit there, the concept of translating whatever kind of basic research outcome a professor can get into a practical enterprise. Start a company is already 10 years ago, it was something that everybody was doing. So coming back to Italy, I was already keen on trying to again translate some of my results, so the results of my research, into a startup. And then you mentioned leaving the comfort zone. Well, what I can tell you is that if you find someone like Lydia leading a company, you keep staying in the comfort zone because she takes care of everything in a wonderful way. So I must admit, I'm not the one that really is getting too much out of my comfort zone. I'm perfectly comfortable as I am.
Thank you, Emiliano. So coming back to somehow to the environment in which we are all interested in, Lydia, Emiliano, can you somehow explain us in simple terms what Sybilla is doing, and at the end of the day, what is the competitive advantage Sybilla may have in this kind of fuzzy and buzzy world of the market?
Okay, do you mind if I share a small presentation?
Please. Well.
Here I am. Okay.
Okay.
It works.
That works?
Yeah,
great.
Okay? See that? Great. So at Sybilla, we work in the early stage of rational drug discovery. What does it mean discovering a drug? It means identify a molecule which is able to bind the target protein and modulate the biological activity. This is what it means. Unfortunately, up to date, a lot of protein targets are still undrugged. Way if they are undruggable because they don't have binding pocket, or they are undrugged because they take on many problems when drugged. So there are still challenges in drug discovery. And here we enter. We want to address the druggability issue. We try to inhibit proteins which are at the moment undrugged. And the way we do it, our innovation, is this new protocol for drug discovery, which enables us to find a completely new category of target proteins. So this is what we do. And why we can do that is because our founders developed a technique that allows us to reproduce the folding pathway of a protein. And this is impossible to do. And so in this way, we are able to characterize, characterize long-lived intermediate state of this folding pathway. And this is impossible to do experimentally because experiments nowadays cannot fully resolve transient states. And it is impossible to do with molecular simulations because it's simply unfeasible. I will show you later some data about that, but it's unfeasible. So with this method that was brought about by our founders, it's finally possible to reproduce the protein folding. And in this way, we are able to find targets in these intermediate states of the protein folding. So maybe Emiliano can tell you something more biologically correct about that.
Yeah, sure. So I will actually just try to be as simple as possible. You have to imagine a protein. Proteins are the workers in our body, in our cells. So are those that really exert all the functions. And there are really hundreds of thousands of proteins that exert even more as a number of functions. They basically take care of every single functional aspect of our cells. What you see in this slide that Lydia is showing is the journey that a protein needs to get in order to reach the form at which it exerts its function. So everything starts from the DNA, from the ribonucleic acids, that gets read. You know that the DNA is basically the book that defines all the information, all the instructions for making our cells and for making our own, basically, for making us living. There are particular machines, they are protein themselves, that read the information in the DNA and they translate it in a kind of different form of nucleic acids, which we call messenger RNA. This messenger word means that they really travel into other molecular machines that translate this information encoded into the messenger RNA into what we call a polypeptide, which is another kind of molecules. Is not a ribonucleic acids, is not a deoxyribonucleic acids. It's actually a polypeptide. So the conjunction of different amino acids, so different kind of molecules. These are the molecules that make proteins. Now, the polypeptide, as soon as it is made, it starts to a process that we call folding. You have to imagine like a chain of little balls, and there are 20 different balls, which means 20 different amino acids, which are the building blocks of a polypeptide. And this chain starts to basically assemble in a three-dimensional fashion in order to make a very unique and precise form. And this form is the one that really specifies the function of the protein. So the protein, after the polypeptide really folds into itself, at the end, it specifies a specific form, and that form can allow the protein, for example, to chunk out other things or to build other things. So basically to exert its function. Now, this is what has been known so far in biology. And so from this standpoint, you may imagine that if you want to prevent the function of a protein, you need to play with these four elements. So either you remove from the DNA the information that makes that protein, which means you remove the gene. In this case, there are particular technologies today that can allow us to do that. But there is, like a con on doing that, because once you remove the gene, you cannot have it back anymore. So it's a very powerful technology. You might have heard about CRISPR, a very widely known these days technologies, which is very powerful, but again has some negative aspect. Other techniques may actually play with the second element, the messenger RNA. They can actually design specific antisense, meaning molecules that really recognize the messenger RNA and block it so that they cannot be translated into the polypeptide anymore. And again, in this case, very useful. As soon as you remove your agent, your antisense agent, you can reestablish the expression of the whatever protein you're targeting. But in this case, it's very difficult to deliver these antisense molecules. So again, it's called RNA silencing, very powerful, but it has some problem. Now, we all know that the vast majority of drugs, when you enter a pharmacy, are small molecules. So small organic molecules that have the peculiarity of being able to travel all around our body and reach the cells that express the specific protein that they want to target. So this is actually something that is very unique of drugs and very desirable. But unfortunately, the first two boxes are not targeted in that way. They are targeted with other emerging technologies, we call it molecular technologies, that still have no matched all the greatness of drugs, of canonical drugs. Now, there is another possibility, which is actually the last box where you see native state. You can actually design drugs, so small molecules that can target directly the protein in its function, in its normal state, functional state, and block it. That's basically what the vast majority, if not all, drugs do. So drugs these days are designed for blocking specific function by binding directly to these very unique conformations of proteins. Now, what Lydia has been talking so far is that there are a variety of proteins very important for diseases that we would like to knock down in some way, ideally with a drug, but we, for some reason, we cannot do it. We're unable to do it. By saying we, I say the entire scientific community. So they're called, like Lydia said, undruggable targets, meaning that we cannot, at the moment, find a drug for them. Sybilla enters exactly in this business because we actually discovered that there is a further step between the polypeptide and the functional native state of proteins, which we call folding intermediates. These are a sort of semi-definitive conformational folding of proteins, but not exactly like the starting point, so the polypeptide, and not exactly like the final point, which is, you know, the protein all folded. There are sort of dynamic intermediate state. Now, we have a technology which is unique. I must say, at the moment, in this precise moment of history, we are the only one that can do this. We can understand, predict how this conformation, so the form of these transient intermediate species is. And at this point, we can capitalize on this information to design drugs for those proteins that usually are not druggable. Why? Because these intermediate forms are different than the native state. It means that most of the times we are understanding now, these days, this thing, they expose some sort of pockets that are not present into the final state, so the native state, and these pockets actually allow us to do something unprecedented, which is basically design drugs that can block them, can bind them and block them. And the last thing, I'm actually getting a little bit long in this, but the last thing is that we realize that by stopping this process, by targeting these intermediate species, then we activate a cellular function that is very widely known that usually recognize these intermediate states of proteins that are stabilized in this case by our drugs and remove it, send it to the trash. So this in fact is a way to silencing the expression of proteins by targeting something that is unique, these conformations, these forms of intermediates.
Thank you, Emiliano. And I must say that you have could have an idea of what is the uniqueness of Sybilla. We have this revolutionary approach, which has been possible because of a multidisciplinary approach to this this problem. We have this is our big challenge, to bring together people from different expertises, from biochemistry to theoretical physics, medicinal chemistry, cell biology, all together, and all with this passion that Emiliano has just shown you. So I don't know you, but I would just stay hours listening to him. Sometimes I ask him for some explanation just for the pleasure of listening to him. You know, this is my great opportunity as a CEO to have this brilliant minds. I must say, any of the founders is really a brilliant scientist. The opportunity I have to lead a team like this is something unique in my life. I think something a CEO would like to have. And when I have a doubt, when I want to understand, because I want to understand one and each step of our processes, I just have to ask. And I'm answer with some preciseness and passion. And this is really peculiar of our team, I must say.
Lydia, sorry to interrupt, really amazing. But looking to your slide, somehow I'm scared. I'm scared because, I mean, I'm reading molecular biochemistry, theoretical physics, medicinal chemistry, cell biology. How did you manage not to fall down an incredible Babel somehow syndrome? It's such a melting pot of different disciplines. Just, I mean, as you said, let everybody understanding each other can be a challenge. How did you manage?
Well, it's our greatest opportunity, really. I don't see it as a problem. I must say, I can understand their language as I am a physicist myself. So maybe the scientific background that I have is easing my pathway among them. Must say so, but it's just it's not difficult. I mean, it's a matter of training it. What we like in our team, also with the young people who has been hired, is that we want to share the information. We are really a community of scientists working together, and we share this information every week. Every week we speak about theoretical physics, molecular biology, cell biology, and over again, it's just about training. It becomes easy to understand each other at the end.
Fine, great.
I don't know, Emiliano, what do you think about?
Well, if I may add, just from my experience, it's a phenomenal opportunity to stay, I always keep saying, in a room where people are talking and you basically don't understand anything. So it's a fantastic opportunity because it's a fantastic opportunity to learn. One of the most recurrent phrases in Sybilla Biotech is that, sorry, I don't understand.
It's me, always.
Yeah, this
always.
Is actually, usually it's Lydia asking, but then me, myself, I mean, like, and everybody else. So we are a mixture of people that I think that our best quality is to recognize that we actually don't know the vast majority of things, but at the same time, we're very curious to studying them. So, and that's the glue that are keeping us together.
And I think that actually your curiosity just produced some results, isn't it? I
guess.
Interrupt you, sorry.
Yeah, I agree.
Oh yeah, that's the explanation, yes.
PPI-FIT, we didn't mention what does it mean. This pharmacological protein inactivation by folding intermediate targeting. So this is the acronym of our model. You want to comment on it, Emiliano?
Yeah, sure. This is actually a graphical representation of what I was saying in the previous slide. As you can see, so first of all, let me make the background information here. What you are seeing here, it's frames of a movie, and we make these kind of movies. Movies of, as you can see, I hope, starting from the left, of this sort of chain that starts to fold. So these are just frames for the sake of, you know, showing in a slide, but we really are really entertainable. I mean, I must admit, at the beginning, when I started collaborating with Pietro Faccioli, the physicist in all this story, I spend my night looking at these movies. I know I'm a nerd on these. I must admit that, but they are beautiful because you see for the first time what I always imagined in my mind. And for the first time, someone showed me a video, exactly a full frame 4K video of how it happens. It's fantastic. So what you see here is basically what I'm saying, the protein folding occurring from the very beginning to the very end. And we get into that information. We get out that information from the movie, from these frames, and use it for drug discovery. That's basically, to make a long story short, what Sybilla does. Pharmacological protein inactivation for silencing the function of a protein by targeting folding intermediates. That's it. You have it, basically everything, all the information in this slide.
Fine.
And as I told you, this is the very first time that we can do it, that the scientific environment can do it. Just as an example, imagine you want to fold a 500 amino acid protein, which is in the bulk of the biological relevant protein size. If you want to do that, and you do it with the most powerful supercomputer on Earth, that was designed specifically to do molecular simulations. So it's not so many computers together, but a computer designed to do that step by step. Then it would take more than a human life. And actually it would take something like 10 lives, living 100 years each life. At Sybilla, with our own small GPU cluster, we can do it in 1 month. So I take something that always Emiliano says, that our is not a quantitative innovation, but it is a qualitative innovation, because we can do what before it could not be done at all, with no means. This is really something impressive. As you can see, the competitive advantage with the other companies that can try to use only plain molecular dynamics or other algorithms to perform pharmacological research, they can only access the millisecond. Now, the protein folding is a process that it may take up to minutes. Molecular dynamics can simulate only 1 millisecond. And now, for the nature of a structural change, such as the protein folding, the system spends so much time waiting for the transition, and then immediately, in 1 microsecond, it does the transition from unfolded to folded. So it means that if you can simulate only 1 millisecond or a few milliseconds, there is really a virtually zero probability that you can observe a transition. You have to simulate seconds to observe transitions. And this is what we can do using the path integral approach, which is a method borrowed by quantum mechanical formalism and so on. So we can factually manage to understand that using the path integral approach could be very useful when looking at transformation thermally driven like this, like the protein folding is. In this way, we can simulate directly the transition, and we can use all the bulk of approximations which were studied for quantum mechanics and adapt them to study this kind of process and to allow a 10 to the 12 dimension integral to be performed. So this is really astonishing. And our competitive advantage, I must say, if one company would start today with what is public, would start today putting together people with different expertise, because it is necessary to do this, to have brilliant people with different expertise, it would take at least, with a team of something like 10 people, at least 1 year to get where we are. But to get to what is published, and it's already 1 year that we are working on it, so we are building up know-how, which is not going public, of course. So I think that this advantage is growing with time. I'm pretty.
So in principle, what you are saying that you are opening up a complete new market for in silico simulation of drug discovery. So is clearly opening a new class of molecules and opening the simulation to a new class of molecules, potentially drug, and to somehow address in silico, so through simulation, in order to have maybe a more effective drug discovery path. But clearly, we are moving in a highly competitive market, in a highly competitive environment. Can you share us what your understanding of this market?
Okay. I have a slide here that tells you how complicated it is. When you buy a drug on the bench, that drug has overcome a process long at least 10 to 15 years. And this drug derives from about 10,000, 15,000 molecules that at the beginning were in the pipeline. So one out of 10,000 managed to become a drug. There are different steps in drug discovery. You have the preclinical approach. Preclinical is in vitro, in cell test, in vitro test, and in vivo on the animals. And if molecules that pass the preclinical after 3 to 6 years, then can go to
Hello? Can you?
Yeah, I can hear. It was
Yeah.
just some scratch.
Okay,
background.
okay.
Can you?
Go,
Okay.
go ahead. Yeah, yes.
Okay. So after 3 to 6 years of preclinical study, you go to the clinical trials that can last up to 6 to 7 years when you test on human people, on people. And the star, the red star over there is what we do. So we found compounds which are in those 10,000. And our aim, of course, is to arrive at least at the 250 compounds which are ready for clinical trials. A few numbers, every year, on average, about 40 drugs are approved. So 40, and you have to understand that those 40 means 10 to 15 years back in time that they started with 10,000 molecules to arrive. Every year, the pharma invest in R&D more than $100 million every year for R&D. Every year, the cost for one drug in preclinical studies and in outsourcing for the pharma, because nowadays the pharma want to do the clinical trials and that's it, and they want to deliver to the CROs all what is preclinical. And what they spend every year for these preclinical studies in outsourcing is about $10 million per drug. So each pharma spends $10 million per drug. And there are something like 10, 20, 30 drugs on the pipeline of each pharma. So it's a pretty big investment. The average total cost for drug, which includes coming from 10,000 drugs and arriving to the one approved drug on the bench, is greater than $1 billion. So it's a huge, huge market. And if we manage, that is what is our aim in the end, if we manage to deliver a molecule which is ready for clinical trials, its value can start from $50 million. I say can start because, of course, it depends on the therapeutic area, it depends on the disease, it depends on many things. But the value starts from $50 million. So we are talking about lot of money.
And then in your opinion, which therapeutic fields you see things are going faster
Well, I must say, of course, there are the undruggable, the still undruggable. They are the one that most drive the market. Of course, cancer research in cancer therapies is one of the most funded. And then there is still engagement in the neurodegenerative research. Also, if it was coming up and down, because it's very difficult to find a cure for neurodegenerative diseases because of the delivery, the problem of delivery. And then we can apply, our method can apply to any therapeutical area. So we don't have to choose one area. I can say that the market value yearly, the B2B2C value is larger than $100 billion for people buying medicines in these 5 areas which are described above. And it's growing every year with a trend of 5 to 10%. So it's a growing market, unfortunately, but luckily for who works here, it's a growing market. And what is important by our point of view, question? Decline. I should decline here, yes, I think so. What is important from our point of view is that more and more big pharma require for CRO to do preclinical research for them. I think that there is some hackering going on.
can you cannot
do you
move?
see that?
No, sorry. No, I cannot see.
I see everything. What's the problem?
Lots of people is requesting remote control of my screen. I'm not
Just
going
forget
to
that.
Forget.
Yes, of course,
Just
but
forget.
it's
People are getting in, guys. I think we need to kick them out.
Yeah.
It's a bombing going on.
Yeah.
See, yeah.
I locked it and I will kick out the ones that are.
Oh gosh, it's exploding.
They're coming and coming now. Oh, wow. Oh my God. Now it doesn't seem to be blocked.
Now we have 99 chats. Yeah, ignore the chat, that's for sure.
It's an characterize, of course, sure.
Yeah.
What shall we do?
I think you should continue because apparently they're not, you know, disturbing the conversation. We may try to
You
go
can
ahead.
just go on. Nobody can talk. So
exactly.
Okay, they're only requesting.
They can only, yeah, they can only make a mess with the chat. But as far as we ignore the chat, we can go ahead.
All right.
Okay.
This is becoming a profession these days. To disturb Zoom meetings.
Anyway, so coming back to our point, also because of, I mean, somehow the time is going on, I see. So maybe a few comments about the model, the business model you envisage in order to attack somehow this market and with this technology. What do you think is more appropriate? I mean, to understanding that we are in early stage of a drug discovery, so pointing to the early stage of, or maybe to the, say, preclinical stage of drug development.
Yeah. Well, what we can do, our in-house pipeline is shown in the upper arrow-like graphs. So we can identify and characterize drug-like molecules and bring them to the red star on the slide before. Mm-hmm. So we can be a factory of those red stars. Mm-hmm. And then we can go on with co-development, of course, with somebody who is interested. But we are starting to think now that it will be probably very important to choose one of these hit molecules and bring it to preclinical. Mm-hmm. And this would really be the major proof of what we can do, and it would add a lot of value to the company. Because of course we are already in the pipeline to do services. We can do services in the early stage of the drug discovery pipeline. We can do software-based services and in silico virtual screening and also in vitro screening. But of course you don't make so much money out of it. So the value of the company grows if we can identify a drug and bring it to preclinical. We are thinking about the possibility of spin-offing on some promising targets. And there is still a lot of discussion about it. Mm-hmm. So if taking Sybilla like a place where a lot of it are built, and then to go deep into the most promising one with other spin-off companies, or if choosing the most promising molecule and going to the preclinical with Sybilla.
I see. Thank you very much. So moving towards the end of this presentation, I will just make a couple of more questions. One is maybe related to our times. Nowadays, we are dealing with pretty difficult times. The pandemic has an incredible impact in disrupting our habits, and affects pretty much all our society. So can you tell us what kind of involvement, if any, did you have with respect to pandemic? In other words, Sybilla did already something to in this respect.
Very much, I would say. We actually were starting working on the COVID emergency on February the 12th. So in the very, very early times where COVID was not yet arrived in Italy, we thought that our duty, having an innovation in our hands, was to try to use this innovation as a service to the community, to try to see. We ask ourselves in one of the Sybilla meetings that we do weekly, oh guys, there is this problem in China. It may be a pandemic, it may become very important. What can we do? Let's study the problem and let's study if we can address part of the problem. And this actually was the case. Nowadays, all of us know very well the mechanism of action of the virus, but it is sketched in the figure over there. The virus has these spiky proteins on its surface, and it uses the interaction between the spiky protein with a cellular receptor, which is the ACE2, which is in our cells. It uses this interaction to enter the cell and start the replication. So we wanted to try to address ACE2 and see if we can prevent the interaction between spiky and ACE2 by lowering the expression of ACE2. So it's what we do. Usually we find a target and we want to lower the expression of the target. So in this case, our target is ACE2, and we tried to target it. And I want to show you this beautiful movie. I'm in love with this movie, maybe more than Emiliano. This is the simulation.
I doubt that.
Yeah. This is the simulation of protein folding, or one trajectory of protein folding. You see that the first structures begin to be built, the alpha helices, so-called alpha helices and beta sheets. And you see that the protein assume a conformation which is more characterized. This is the intermediate state. In this state, you see the shaded violet area. It's the druggable pocket that we can characterize. Inside this druggable pocket, we try to find molecules that can bind. So there is a virtual screening of molecules. And once we find molecules that can bind, we bring those molecules in the laboratory. Because so this is in silico. If we remove the molecule, you see that the protein continues its folding and gets finally to the native state. Now it's just, you can see the helices blop doing this and get
it is not any more druggable, isn't it?
In this way, it's no more druggable because the helices just close the door.
So meaning,
yes, this is important to say, because the druggable pockets that we can find in the intermediate state are not present at all in the native state.
Okay.
And are also kinematically far from the native state. It means that there is an energy barrier that the system has to overcome to get from the intermediate state to the folded state.
Great, incredible, really amazing.
Yes, so this is the sketch of the intermediate state, and I must say the way it was reached, this information. ACE2 is the largest protein that we have ever simulated. It consists of 600 proteins. It's huge, huge. And it's difficult it's feasible to study the folding of such a protein, but with our computational stuff, we would have taken something like 5 to 6 months. And we thought, we tried for 1 month, and we only got 2 trajectories. So we understood that 6 months were due in order to have some results. And we thought that this could not be right in this case of emergency. So we asked to the Italian National Institute for Nuclear Physics, which has all the supercomputers that were used to find the Higgs boson at CERN, for instance. So they have a lot of facilities, and they immediately repurposed most of their Italian facilities to work, to let us simulate this protein. So we use something like 30,000 cores in different data centers. And it took, the simulation time was just 2 weeks. Just to make an example, if you use one Pentium computer, which has got 4 cores, you take 160 years of computation. So really we were allowed to use a huge amount of computational facilities. It was a very nice example of cooperation in case of emergency. So we could characterize the folding intermediate of ACE2 and also the binding pocket. I like this slide because it can show you very well the peculiarity of our approach. So here you see the molecule, the protein with the shaded violet area, which is the druggable pocket. The green helix in front of the druggable pockets belong to the native state. So it's not there when the system is in the intermediate state, but it shows you how the native state actually block the existence of that druggable pocket.
Mm-hmm. Great.
So we made virtual, we had to choose which molecule to screen, because usually we use a 10 million molecule library and then we try to find the best one that bind better to this druggable site. But in this case, we couldn't afford the 10 years of process for getting a drug. We wanted to get a cure for COVID. So we performed virtual screening only on FDA approved molecules or molecules which were already in clinical trials. They are something like 9,000, the ones who are commercially available. And out of those 9,000, we found 35 molecules binding in silico, the intermediate state. 17 of which were already FDA approved. That means that you can find them in the pharmacy on the bench, and other 18 are investigational. And with our great excitement, Emiliano, you want to comment on this?
Well, I think very quickly, because we really are running out of time. To make a long story short, we validated all our predictions that at the point of this slide were just in a computer. So we're just really guessing that we were really having an impact on this protein. Well, this was translated in our lab into cells. So we were basically able to demonstrate that we are capable of exploiting our technology in cells that express the receptor. So that's so I must say, in total absence of intrinsic side effects. So in terms of toxicity of the compound that we found. So at the moment we have a drug that is used in a completely different setting in the clinics. It's an FDA approved drug. So used actually since 15 years ago. And now we want to make a sort of repurpose. In fact, it's exactly the word, repurposing this drug for suppressing the expression on the surface of the cells of the receptor for the coronavirus. And then hopefully this is actually gonna somehow hijack the ability of the virus to replicate. But to really make this point, we will have to demonstrate it again. So this is just a guess for the moment. We have solid, let's say, hypothesis for guessing that we are going to be able to do that, but we have to prove it. So in fact, we're trying to collaborate with hospitals in a way that, they have access to security levels that allow them to work with the entire virus, with infectious virus, in order to test our compound and really demonstrate that our compound can inhibit the replication of the virus. And if so, we might actually have an opportunity for translating our research into the clinics again. So really trying on patients.
Amazing, really amazing. Thank you, Emiliano. So really getting to the conclusion, a single, sketch about what you see in the future. What would be the future for Sybilla? Really quickly, do you see any technology which may impact Sybilla? Or do you see any what will you see the results of all this development time?
I think I will answer first, just to make, you know, like the more romantic side of the story. What I truly hope that is that Sybilla is going to be able to cure a disease. At the moment, I cannot tell you for sure which one, but I'm very optimistic that with the kind of love that we're putting into our effort every day, and we have a fantastic team. I used to say, if you knock the door at the Sybilla office, all you find is not scientists or business or whatever, you find people in love with what they're doing. And this is actually the magic element that we are putting into our company every day. So if you ask to me, it's more a dream than what I really rationally conceive, is that we're going to be able to find a cure for a disease.
Thank you, Emiliano. Amazing.
You are the romantic, Emiliano. I'm the practical, you know.
Yes, exactly.
I want to.
Which are both necessary, by the way.
Both are necessary, of course. I use want because really I want to be cutting edge in technology, keep on being cutting edge in technology. Now we have these fantastic disruptive results, and we don't want to leave behind quantum computing and artificial intelligence, of course. We think they are the next interlocutor, the next arguments that we have to deal with. Yeah. And we are already working to see the pipeline going in that direction. So I really would like to have a company which is always cutting edge in technology.
Thank you, Lydia. Francesco, do you have a question?
Well, actually, Lydia answered before I asked the question. Because it was exactly that.
Oh, she always does that. That's typical.
That's amazing.
But I know that we are late, but I have to show the last slide because it is our strength. Really, sure. All the people working in Sybilla deserves to be mentioned because it is the best team that a CEO could think about. They are really passionate, working passionate with love and passion and with competence. Really competence, and we are growing these young people in the bottom line, and they are they range from theoretical physics to quantitative biology, and they really are growing in this multidisciplinary environment, being part of a community. This is really our challenge and our strength, I would say. So we cannot, I always have to say that because they are really, really fantastic. It's an amazing group.
Great, Lydia.
Yeah, I want to just to add to this that there is a feeling that actually, more than a feeling, I'm very convinced that in an academic lab, as far as also in a company, the best situation is to recruit people that are better than you. So than yourself, and we are absolutely, this is absolutely true in Sybilla. The best people that you find are the youngest. So lots of excitement for that, because you know, even if we don't achieve what we want, there is very high chances that they will achieve it.
Thank you very much, Lydia. Thank you very much,
guys.
Emiliano.
Really great story, great company.
Keep your passion, keep enjoying your work, and keep growing your team. All the best for the future.
No problem with that.
We will, yes, exactly.
I hope so.
We know you hope so.
Thank you very much.
Okay, thanks a lot, guys.
Okay.
Thank you very much.
Thanks for sharing. Goodbye, good night.
Bye,
bye.
Bye.
Thank you, everybody, for coming, and see you next time. Bye-bye.
Bye.